So next up, we have Dr. Richard Judson. Richard, if you want to go ahead and start your camera, and you can start sharing your screen too. Richard is with the US EPA, the Center for Computational Toxicology and Exposure, where he develops new methods to evaluate chemical safety without using animals, which we appreciate. His area of expertise is the development of computational models of biological systems. He has his bachelor's degree in chemistry from Rice University and a PhD in chemistry from Princeton. And Richard's fun factoid is that his daughter is an archaeologist who is supposed to be working on Crete right now, but because of COVID, she's had to be living at home. And for his sake, fortunately, his daughter and his wife are competitive cooks, so he's spending COVID eating very well. So thank you so much for joining us today, Richard, and the floor is yours.
So like Tom, I still play the fife, and I was doing that COVID time until I broke my arm. So we're both now non-players. Okay, so thank you for having me speak, and Tom actually did a good introduction to a lot of the things I want to talk about, so I can skip over some details. Okay, so we all are aware of the background of the oil spill. So I'm with the Environmental Protection Agency, and I'm in the Office of Research and Development. But what we do is we provide the science basis for the regulators to make their decisions. And why we're involved with this, there are a number of reasons. But when there is an oil spill, the EPA, and it's actually the administrator of the EPA, is the one who gets to decide whether you get to use the dispersant and what dispersant to use. And so Gina McCarthy was pulled into this early on in the spill, and there was a lot of concern about using the safest dispersant that you could use. And so we were asked to very quickly evaluate a range of dispersants and see, was there one that was less toxic, excuse me, than Corexit, which is what was being used initially. The laws that govern how we get involved with all of this is the Clean Water Act and the Air Oil Pollution Act. And the main things I want to point out here is there's what's called the NCP, the National Contingency Plan. So if you're a company and you want to have a dispersant or a surface washing agent or any of these other kinds of cleanup products that might be available to use, you have to do a certain amount of toxicity testing. You then put it on the list, and then it becomes a candidate to use. Being on the list doesn't mean you can use it. It means that the EPA could approve it for use. Okay, and so a number of companies had submitted dispersants to the list. They were all on the list, and the EPA could approve any of those in principle for use. And so we did testing across. There were actually 8 of these we tested. Okay, so Tom did a good introduction to what a dispersant is. Couple of points here I want to make clear. So he kind of mentioned this, that the Corexit formulation was not public when all of this started. They ultimately provided that to help people feel a little better about it's being used. But at least when we got started, the whole set of 8 dispersants we were asked to look at were black boxes. They were just this witch's brew, as far as we knew, just witch's brew of nasty stuff. And we needed to just test them to say, is one more toxic than the other, and so on. The major part that Tom mentioned are these detergent aspects, but they have some other components in there. They have colorants, so you can actually tell one dispersant from the next. They have their different colors, and I'll actually show that. Okay, so what distinguishes one dispersant from another? So obviously efficacy is what you worry about. How well does it disperse the oil and make it available to be biodegraded? The EPA doesn't regulate on that, so that's really kind of a customer issue. They want something that works well, and I suspect they all work about as well. What we do regulate on is toxicity, and it's really how much of the dispersant is required to be toxic. And Tom was quoting numbers in parts per million. So this is, you take a volume of dispersant, so a gallon of dispersant, and you dilute it in a million gallons of seawater, Gulf water, that would be 1 part per million. And the toxicity testing is there are 2 species. There's the silverside minnow and the mysid shrimp. And there's a standard test where you have a tank that has these creatures in it, and you put a certain amount in and say how many of the fish died, and you put a little bit more in and how many of the fish died. And what you do is you finally come up to the concentration in parts per million where half the fish die. So that's the lethal dose 50 or the LD50. And that's what everybody, all the companies who submit these report. And so on the list, the National Contingency Program list, we have the LD50s for these 2 species for all of the dispersants. There is this special concern, and I'll get into this. This is really what we were focusing on, is that some of these dispersants were potentially estrogenic. So they had compounds in them that behave like the natural hormone estrogen. And I'll tell you why that's critical. So this down here at the bottom, this chemical, this is nonylphenol ethoxylate. So it has a benzene ring, it has 9 carbons at one end, and it has what's called this ethoxylate group. So an oxygen, carbon, carbon, oxygen, and so on. If you break the bond next to the oxygen, it starts to look very much like, at least to the body, it looks a lot like natural estrogen, the hormone estrogen. We had no way to know whether any of the dispersants, no official way to know whether any of the dispersants actually contained nonylphenol ethoxylate. And so we had to simply test them to see whether they behaved like they had estrogen there. So we all know estrogen is a natural hormone. It's required for sexual development. A lot of processes throughout your life. We think about estrogen as the female hormone. Women have more estrogen than men, but it's, men have some estrogen and it does important things for men as well as women. If you expose an animal at the wrong time or at the wrong level with an estrogen, you can have all sorts of bad things happen. So one effect that we see quite often in waterways downstream from wastewater treatment plants is what's called intersex. So male fish start to develop female characteristics. And that can, of course, lead to infertility, at least all sorts of other problems. In humans, inappropriate exposure to estrogens can have lots of problems. The whole issue of worrying about endocrine, environmental endocrines came about with diethylstilbestrol, which was a morning sickness drug, which was used in the 1950s and 60s. And what happened was the daughters, the daughters of women who took this drug while they were pregnant, when those daughters were in their late teens, early 20s, many of them developed what had previously been very rare, uterine cancers, breast cancers, reproductive tract cancers, and most of them were infertile. And so it was kind of like a ticking time bomb. You dose a baby in utero, a fetus in utero, and 20 years later you see these very bad effects. And so EPA has developed a strategy to test chemicals to avoid this sort of thing. And so in particular for the oil dispersant issue, the worry was you would have this, some kind of an estrogen would then float into the breeding grounds along the coast in Louisiana and Texas and Mississippi, and it could cause a crash in the populations because you decrease fertility. And you have to balance the risk of whatever the dispersant might be from the known effects of having actually that oil slick wash up into the breeding grounds. Okay, so the studies we actually carried out was we reran the standard toxicity studies, the minnows and the shrimp, just to confirm that the companies who submitted to the product schedule had done a good job. And we actually saw there were no significant differences. And in fact, the EPA studies made things look a little safer than what the companies had said. There was another concern that if the dispersant mixed with oil would be more toxic than either the oil or the dispersant itself. And so these were studies done by our lab we have a lab on the Gulf Coast in Florida. And again, they found no significant difference that, adding the dispersant to the oil or the oil to the dispersant is not more toxic than either of the others. And then what the group I'm part of did was we ran a bunch of studies in human cells. So we didn't use real humans, we used human cells. And so what we wanted to know is how much of the dispersant is required to actually kill cells, which is called cytotoxicity. Is there any estrogen-like activity? And then we looked at a number of other features. So is there testosterone-like activity? And I can just say right now, we didn't see any of that at all. Okay, the dispersants we looked at was obviously we looked at Corexit, which was being used initially in the Gulf spill. And my understanding was just because there was a lot of it available there in the area. And so it was the only business in town initially, but then these other 7, the companies, the products applied to be used. And so the EPA administrator just to evaluate all of those. Okay, so you have to suffer through a little bit of molecular biology here to understand what we did. So how estrogen or estrogen-like compounds work is they actually bind to a protein, which is called the estrogen receptor. And so I'll just use this abbreviation ER. And when a chemical, a hormone binds to the ER, it actually causes certain genes to be turned on in the cells. And those genes, what they do is they cause the cells to start proliferating. So they divide and they grow. And so for instance, when a female creature, any mammalian female goes through puberty, their ovaries start producing estrogen, then that will cause the uterus to mature and expand. So this is the normal process of what's going on. And people, very clever people working in the labs have developed ways to engineer human cells so that when the chemical binds to the estrogen receptor, something happens, you can measure the cells turn green. So that's the sort of assays we run. And they're called in vitro assays. They're run in little glass petri dishes. And just a little bit of how we actually do that. We have these plates. They're actually today plastic instead of glass. And these are about 3 inches by 5 inches with a bunch of little wells. And you put cells, human cells in each of the wells, and you add chemical. And then you use big robots or use little robots. And this is my colleague, Katie Paul Friedman, who runs little robots and does lots of stuff in the lab. And you essentially say, okay, did the cells turn green? Was it estrogenic or not? And what you do is you always run over a series of concentrations. And this is what good data looks like. At low concentration, you don't see anything, and then at high concentration, you do see something. So that's basically the data we generate for the dispersants. And we use 3 different technologies. The first one was you can actually run this without cells. You just have the human proteins and see whether the chemical binds. Turns out that this detergent part of the dispersant simply destroys proteins, at least high concentrations. So those assays didn't work. And then we ran a couple of other technologies, one at a lab up at the National Institutes of Health. They're the ones with the big yellow robot I showed on the last slide. And then there's a company in Research Triangle Park here called Adagene. And in both cases, we looked at estrogen and cytotoxicity. And so this is what the data looks like. And Tom made the point that a low number, so this is a concentration of the dispersants in parts per million. So he was quoting parts per million numbers. A low value is more toxic. So something that kills cells, and this is just this kill celling I'm showing here, very low concentration would be more toxic. And something that takes a lot to kill the cells is less toxic. And so we see here Corexit is the 3rd from the bottom. And so it's kind of the middle of the road cell killing ability. So it's certainly less bad than some of the others. Lots of points here, but look at these, the gray and the black triangles. Those are the concentrations where the fish and the shrimp are killed. And so the fish and the shrimp tend to be a little more sensitive than the human cells are. Okay, but it's really kind of in the mix. But it's also about the same level. And so by looking at what happens in human cells, you can predict what's going to happen in the fish and the shrimp and vice versa. Okay, but the major point here is that the Corexit is certainly no worse than a lot of the other for killing fish and shrimp. And notice the magnitude of these numbers. If you remember back to Tom's slide, things quickly get diluted down to 1 part. This is 1 part per million. Here, you have to get 100 times that to start seeing any sort of cell killing or fish killing or shrimp killing. Okay, the endocrine activity, estrogen. So we didn't see any androgen receptor activity. Nothing looked like a testosterone derivative. We saw really weak activity for estrogen in a couple of the dispersants, so Nokomis and Sebrat. You know, initially I had no idea which one of these might or might not contain that. The internet's useful. It's a great thing. There was a little blurb from the CEO of the Alabaster company who makes Sebrat. They got taken down not long after I found this. It said Sebrat contains no more than 2% of this nonylphenol ethoxylate. So he was essentially admitting that Sebrat had that. And Nokomis, who makes the Nokomis 3F4, said that they will make you a formulation without nonylphenol ethoxylate, implying that the standard ones that they had out there probably did. So we felt like we had probably correctly measured that these two did contain the nonylphenol ethoxylate. Just a little bit more biology. We tested lots of other kinds of biology in our cells, and just one little factoid here. So these yellow dots, again, this is the 1 part per million, 100, and so on. These are genes that are associated with detoxification. So what your liver does, your liver is the first line of defense against toxic chemicals. So if you are exposed to something toxic, the liver turns on enzymes that break it up. And these yellow are associated with particular, essentially they're detecting there's something bad going on in these cells. So this is kind of the right thing you want to do if you were exposed, if cells were exposed to the dispersants at relatively high levels, again, higher than you'd probably be actually in the diluted Gulf of Mexico, your liver would detect that that's there, and it would arm itself to detoxify. Okay, so in conclusion, we saw some weak evidence of ER activity in a couple of dispersants, but nothing in the Corexit, and Corexit's kind of in the middle of the pack for cytotoxicity, and the EPA recommended continued use. And the most important thing here, again, going back to comparing with the numbers Tom was talking about, any kind of activity, whether it's estrogen, cytotoxicity, killing fish, shrimp, upregulating liver enzymes, all of that happens at hundreds to thousands of times higher concentration than you would see after a few hours of mixing all of this stuff into the Gulf of Mexico. And certainly by the time anything got to the breeding grounds along the coastline, there would be an infinitesimal amount, and so almost certainly no effects at all. And so finally, I'm part of a big team at EPA, and just want to mention folks, and here's my email address. If you have questions, feel free to send me an email. And I'm done. Thank you very much.